Hi, welcome everybody. We'll give it a couple seconds to let people settle in here. Great. Well, as more people file in, I will start our webinar. Welcome everyone. Thank you for joining our science briefing titled Ask an Expert: Advancing Next Generation Ocean Management with Environmental DNA. My name is Tanner Waters, and I'm a Science Policy Fellow with the California Ocean Science Trust. For those unfamiliar with us, we're an organization that strengthens the bridge between scientific research and sound ocean management. OST was created by California state legislation to bring world-class science and innovation together with state and federal policymakers to accelerate progress toward a healthy and resilient coast and ocean. As a nonpartisan organization, OST serves a unique role in supporting the California legislature and state agencies in the stewardship of our ocean resources. This briefing is brought to you by OST's Legislative Science Service Program, which brings science advice to lawmakers. If you enjoyed this one, we do this about once a quarter. Our other recent briefings have included kelp recovery, ocean noise, ocean and coastal impacts of wildfires, and harmful algal blooms, which can all be found on our website. Before we move into remarks, I'd like to introduce the sponsor of this briefing, Assemblymember Papan. Assemblymember Papan represents the 21st Assembly District, which includes the entire Bayside of San Mateo County. She received her BA in political science from UCLA and a JD from the University of California, Hastings College of Law. Previously, she served as mayor and city council member for the city of San Mateo, and in the assembly, she serves as the chair of the Assembly Committee on Water, Parks, and Wildlife. From all of us at OST, thank you so much for sponsoring this briefing and for all your work on environmental issues. I'd like to now invite Assemblymember Papan to unmute and turn on her camera to kick off today's discussion with some opening remarks.
Well, thank you so much, Tanner. I'm just delighted to be with you. As you mentioned, I'm chair of Assembly Water Parks and Wildlife, emphasis on the water, so it and wildlife. So it does have a very special meaning for me and the work that I'm doing in the Assembly. And my hat's off to California Ocean Science Trust for bringing us together for what I consider to be a very important conversation about environmental DNA. And I hope we can put this tool to work in ocean management. For those of you who may not know me, I'm all about proactive management and data, and I believe in anticipating problems before they become crises. So, using the best science available to guide our decisions is always something that's very appealing to me, and that's why I think eDNA is really so compelling. Um, so, eDNA really does allow us to manage better than we ever have in the past. I think. The fact that it's just a way of really offering real-time intelligence about what's happening in our waters cannot be underscored. Um, Trying to figure out which species are present, which are declining, which invasive species may be quietly gaining ground, often long before surveys really detect anything, is absolutely game changing. Our oceans are changing, they're warming. We see a lot of shifts in currents and food webs, and things that once thrived may no longer thrive, and what couldn't survive before all of a sudden may be very welcoming in a new environment. So I can't say enough about the idea that eDNA is really going to allow us uh, to stay sort of ahead of the game and to be proactive. And just like we're seeing with the golden mussel, any delay in detection can really make a problem much more difficult than we would like it to be. So, and cost us a lot of money. So anyway, I'm excited about eDNA because I really think that it's going to help us make smarter choices. It's going to help us make every dollar that we can contribute to certain research and operations go as far as it possibly can go. And we can also protect vulnerable species in the meantime that may be on the brink. So I'm thrilled to be with you today. Thank you for giving me the opportunity just to sort of open up. And I look forward to turning you over to those that are really going to tell us what it's all about.
Thank you so much. And I think you nailed it right on the head, exactly what eDNA can do. It's not just getting better information, it's being able to do that information, get that information earlier so we can make those decisions before problems become real problems. So thank you, Assemblymember Papan, for those remarks and for all your leadership on coastal and ocean issues. Um, Next, I'd like to introduce our panel of experts who bring a wide range of scientific and management perspectives on how eDNA is being used to advance our understanding of oceans. Panelists, please feel free to turn on your videos and join me. Welcome, everybody. First, I'd like to introduce our panelists. We have Dr. Susie Thoreau, who's a principal scientist at SQUIRP, or the Southern California Coastal Water Research Project, where she leads the environmental DNA research portfolio. Her work focuses on developing molecular methods for biomonitoring and bioassessment. Dr. Thoreau also leads the California Molecular Methods Work Group, a work group under the California Water Quality Monitoring Council, which is focused on optimizing and standardizing eDNA approaches for informing management decisions. Next, we have Dr. Ryan Kelly. Trained as both an ecologist and a lawyer, Dr. Kelly has a broad set of interests focused on hard scientific data and policymakers' use of those data. He's the director of the eDNA Collaborative, an associate director of the School of Marine Environmental Affairs within the University of Washington's College of the Environment. He's a California native with a bachelor's degree from UCLA, a PhD from Columbia University, and a JD from the University of California, Berkeley. Next, we have Dr. Kevin Yamahara, who is a research specialist in the Surf Center at the Monterey Bay Aquarium Research Institute, or MBARI, specializing in biomolecular approaches to understanding and protecting marine and freshwater ecosystems, with a particular expertise in omics-based technologies and real-time environmental monitoring. As a leader within the Ocean Biomolecular Observation Network, the Marine Technology Society's eDNA Committee, and a founder of the Marine Omics Technology and Instrumentation Workshops, he works at the intersection of engineering and molecular science to advance next generation sensing tools for monitoring aquatic ecosystems. Last but not least, we have Dr. Zach Gold. Dr. Gold is the Ocean Molecular Ecology Lab Group Lead at the NOAA's Pacific Marine Environmental Lab in Seattle. Dr. Gold received his BS from Stanford University and PhD from UCLA. His research seeks to leverage the advances in molecular biology to scale biological analysis with physical and chemical processes and allow for the characterization of marine ecosystems' response to climate change. And our moderator today is Dr. Melinda Baerwald. Dr. Baerwald is an environmental program manager in the Division of Integrated Science and Engineering at the California Department of Water Resources. She manages the Collaborative Science and Innovation section and co-leads DWR's Genetic Monitoring Lab, which focuses on applying genetic tools like eDNA to protect the San Francisco Bay Delta ecosystem and reliably supply water for California. A big thank you to all of our speakers today. I'm really excited to get to hear from you all on this important issue. For our guests, the structure of this panel will start with 15 minutes of introductory questions to get to hear from each of our panelists. Then we'll open it up to questions in the Zoom Q and answer function and those that were submitted when you registered. So please feel free to submit questions as they come to you. And with that, I'll pass it over to our moderator to open up the panel.
Okay, great. Thank you so much, Tanner. We're going to start with discussion about some basics behind environmental DNA. Zach, can you explain what environmental DNA is, how it works, and how confident researchers are when they detect eDNA that the organism was really there?
Yeah, absolutely. Happy to answer that, Melinda. So environmental DNA or eDNA, can be found in the environment, whether that's water, soil, or air. And this is DNA that incorporates whole organisms like bacteria and phytoplankton, as well as shed cells and free-floating DNA that might derive from organisms, whether that's mucus or skin or feces or scales. And a lot of recent work is showing that it's largely whole cells, particularly from larger animals. And just like we shed skin and hair, when we are in the shower, organisms in the ocean are constantly shedding their DNA, whether you're a crab or a whale or a fish. And so we can go out in the environment and we can grab a water sample, we can filter it down, then we can take it back to the lab and do our analysis, or in some cases do that in the field and characterize what animals and reconstruct who is living in the environment from the DNA they leave behind. There's three kind of main approaches to eDNA. There's single species approaches. These are often more quantitative and more near real time. And so we're looking for one target or maybe a couple targets at a time. And these are, very similar to the COVID tests that we all had to take during the pandemic. There's sort of meta-barcoding approaches where we can look at multiple species, hundreds to thousands of species at a time, and lets us really characterize a whole community of things we're interested in, maybe fishes inside and outside a marine protected area, for example. And lastly, there's a shotgun sequencing approach, which is not targeted. We're just going to sequence all the DNA that's out in the environment. And that's usually really helpful for telling us from, especially the microbes, what their functions are in the environment and what kind of metabolic processes are happening. And so depending on the research question, you might want to use one or all three of those approaches that you're looking at. And then to answer your question about, what does an eDNA signature look like and what is it telling us? Well, we know that the amount of DNA that we collect is a function, of the biomass and the number of individuals that are out in the ocean. It's also, a function of the shedding and decay rates of different species, and then that fate and transport in the environment. But really what we know is that eDNA signatures are biased to who is here and most recently and the most abundantly. And so we, over the last decade, spent a lot of research, some of us on this call and many, many others in the field, looking at, putting some bounds on how long eDNA lasts in the environment. And it really looks like it's on the scale of hours to days. And in general, in marine environments, the eDNA isn't transported too far. We're talking about on the scales of meters to maybe kilometers, some sort of extreme cases, with really strong currents or really strong tides. But in California, we've seen that you can tell differences on the microhabitat scale, 5 meters within a kelp forest, or, the difference between inside a kelp forest and sandy bottom, 10, 50 meters away from each other. And so it's really, giving you a snapshot of who's there, here and now. And, to go to the trustworthiness, I think what's really been amazing over the past, 2 decades was really we've gone from this almost seems like magic or a black box that you can go out in the environment, take a water sample to, oh, this is routine. It's really easy to implement. And we've learned a lot about, how and when we can deploy eDNA so that we trust the data we're getting back. And so, really across hundreds of papers in the marine environment, we consistently see that, well-designed and well-validated eDNA surveys outperform a lot of the old school manual, traditional surveys, and particularly for, really cryptic or really rare species that are often, hard to find or difficult to survey for. And, the other thing about, trustworthiness of the data, eDNA, is quite cost effective as well as being really sensitive. And so that, lends itself to more replication. We can go out to a site more often, take more samples, increase that replication. And so that, obviously, as in all kind of observations, provides more trust in the data, that we get a multiple detections, multiple data points, to confirm that. And then as well, one advantage is, you can archive an eDNA sample, and it lets you go back in time to revisit those samples. And so if you get a detection that, maybe you weren't 100%, sure of the first time around, you can rerun it, a dozen times if you want to really confirm, the results that you're seeing. And so, as a mentor friend of ours, often says, Ole Shelton, that eDNA is really just a new net of a new way of observing the marine environment. But it's really powerful. And so just as an example from our own lab, in just the last 2 years, we've, processed about 12,000 eDNA samples. Our estimate across the whole West Coast is something to 20,000, and that's given us a really good idea of, how, you can trust the validity of eDNA surveys, but also the scale of what's powerful. So like our team of 6 with these 12,000 eDNA samples detected over 50,000 species. I mean, this is mostly microbes and phytoplankton, but, hundreds of species, of fish and marine mammals. And that's the equivalent of about an observation of 2 species every second for the last 2 years. And so the scale of that is what iNaturalist was in 2020. And so, we don't have another technique, certainly not with binoculars, certainly not under a microscope, where we could possibly ever think to make, that many observations per second. And so in the same way that remote sensing has really revolutionized, how we view the Earth's surface, eDNA is giving us an opportunity to look at the biodiversity at a scope and scale that we really have never had the capability to do that before. And being able to take more samples over more time, over more depth, over more space, with often much higher taxonomic resolution, because the DNA can get us down to species or often strain level. Lets us understand our marine life and our marine environment like never before.
Thanks so much, Zach. That was a wealth of information, and I found it really helpful for myself. And I would say one thing that stuck out to me was, you mentioned that you could go back in time, and if you needed to repeat samples. Assemblymember Papan brought up the golden mussels, and that's something that we can do as well. Like for samples, even before we knew it was here, we could go back if we have archived eDNA samples and bring and check for when was it really here, because it's such a sensitive tool to be able to figure these things out. So that is great. Thank you so much. Okay, so the next question is for Susie. Susie, eDNA can be applied to a number of use cases, from fisheries management to invasive species, such as golden mussel, offshore wind monitoring, and water quality testing. How has California incorporated eDNA into ocean management, and what lessons have come from these efforts?
Great. Thanks, Melinda. So as Zach just gave you a preview, there's a number of applications or use cases where eDNA is being incorporated into routine biomonitoring or bioassessment, and in some key instances is actually being used to inform management decisions. And so highlighting, I think, also one of my favorite examples of where agencies like the California Department of Water Resources and California Department of Fish and Wildlife are using eDNA-based tools because they are so sensitive to look for those needle in the haystack, so invasive or endangered species such as the quagga and zebra mussel, or as the recently discovered golden mussel in the San Francisco Bay Delta region and now throughout California waterways. For these instances, it's critical to have that very sensitive tool like eDNA to be able to detect the organisms when they're in really low abundance so that we can institute a management action, obviously before the problem gets worse. Some of the other eDNA applications, like Zach mentioned, looked instead at whole communities of organisms. So maybe we're looking at entire fish populations. We want to know all the species that are present there, or maybe entire harmful algal bloom communities. One of the highlights that I wanted to focus on was work being done by the Cal Coffee program through NOAA and the Scripps Institute of Oceanography, where they've been demonstrating the power and potential of using DNA metabarcode sequencing to profile those entire larval fish communities as well as zooplankton communities. And as we heard, some of those samples are coming from net tows that were taken decades ago, but because they are preserved in ethanol, we can go back into that archive and be able to reconstruct those biological communities. So another key advantage of these eDNA-based tools. I would say one of my other favorite examples of where we're using these molecular methods or DNA-based approaches to inform management decision-making in California is in the use of targeted assays for quantitative PCR, or now it's close analog digital PCR, for beach water quality monitoring. So down in San Diego County now uses molecular assays to look for the presence of human pathogen bioindicators or enterococcus bacteria. And then they will use that information to make beach closure decisions based on the number of gene copies of those bioindicators that they find present in the water. So here, the real power of using eDNA is, again, that you can make those decisions in a really timely manner, on the order of a few hours of sampling the water, instead of previously having to wait maybe a few days using traditional or culture-based methods. So in my opinion, across all of these different use cases and applications, one of the biggest lessons learned about eDNA and its relevance to management is the importance of co-design. So having decision makers or managers or end users involved in the research and the piloting of these tools as early as possible. So when we're onboarding a new technology, it's really helpful to have those managers at the table when the study is being designed, through when the samples are being collected or analyzed and interpreted. I think this really helps build the trust, and understanding the resulting data helps us understand those strengths and limitations and identify any future potential roadblocks for future adoption. Thanks.
Thank you so much, Susie. And I couldn't agree more about the managers. Speaking as someone that is kind of in between being a scientist and a manager, I find that scientists don't always speak the same language as managers, and understanding what managers' questions are and then how you can use the scientific tools to help them. You never want to create problems for managers. Instead, you want to help them solve their problems. And so, great points. Thank you. All right, Kevin, eDNA is often described as a next generation technology. How does eDNA integrate with newer technologies like autonomous deployable instruments, robotics, and artificial intelligence?
That's a great question. Thanks, Melinda, for the question. I just wanted to follow up on something Susie said and you also mentioned, Melinda, on this idea of talking different languages to different groups of people. And so while you guys were talking about scientists to managers, why I'm increasingly discussing with folks or the conversations I'm having are from engineers to scientists. And so if you're developing instrumentation, we're often seeking information from scientists on what they want and what they require for their instruments that they want designed. And I think so there's a whole range of discussions that need to happen with the co-design of instrumentation, but also the co-design of how we implement eDNA tools in general. So I think that follows into the thread that I'm sort of going to talk about right now. And I think for new technologies that also are developing in parallel with all these eDNA methods is this idea of autonomous systems for eDNA sampling. So for environmental sampling, being there is everything because you can't measure what you never capture. And I think this is where eDNA and innovations in marine robotics sort of advance our understanding of the oceans. So environmental DNA is often described as next generation technology because it fundamentally has transformed how we detect, monitor, and understand biodiversity. But I think the real power of eDNA is realized when we begin to integrate those with other advancing technologies such as autonomous samplers, robotic platforms, and then artificial intelligence. So this creates a tightly coupled system for scaling real-time ecosystem observations. So together, combining all these technologies, we shift eDNA from specialized lab methods and monitoring methods into an operational sensing capability. So advances in autonomous and deployable instrumentation are transforming eDNA into real-time biological sensing capability, and that these new generations of samplers can perform in situ filtration, preservation, extraction, and even in some cases onboard molecular assays like quantitative PCR or sequencing. So by operating independently of ships, when we couple autonomous sampling systems with autonomous platforms, these systems can provide unprecedented spatial and temporal coverage of that traditional ship-based sampling can't achieve. So it also enables this idea of continuous observations in our marine systems. Moving a little bit further from that, I think we also coupling these systems together with eDNA, we also support event response sampling. So instruments positioned in dynamic habitats throughout California, such as estuaries or upwelling zones that can capture really short-lived processes like harmful algal blooms, migration pulses, and spawning events, or these early stages of biological invasions that Susie was mentioning. So the advances in marine robotics are dramatically expanding where and how eDNA can be collected. So the integration with all of these platforms makes it possible to sample in other locations that were likely otherwise inaccessible, like deep sea vents, submarine canyons off of California, seamounts, under ice environments, and lots of other challenging habitats. One thing that I'm really excited about with all of this technology coupled together is the possibility of using AI and machine learning to transform eDNA into real-time ecosystem intelligence. So AI might enable intelligent sampling by guiding when and where autonomous systems collect eDNA, by using predictive models that detect environmental anomalies or forecast biological events based on other types of signals, whether that's fluorescence or acoustics or temperature sensors. And then this creates adaptive sampling systems in which autonomous platforms can dynamically reposition themselves or intensify their sampling efforts in response to these different emerging conditions. Yeah. think that's all I had to say.
So much, Kevin. And I will tell you that, it's often said with different molecular tools that there is no silver bullet, but the use of autonomous sampling almost feels like it almost is in terms of the capability of what it could do. And I will tell you that our executives at the Department of Water Resources, when we've discussed autonomous samplers, the ability that we don't have to bring crews out there, for instance, in inclement weather at night, because we do sampling at these times, that safety is a real concern. Having an autonomous sampler out there, and then the possibility in the future of them being able to get like a remote ping sample of saying, oh, your target is now where we were looking for it. For instance, for us, the State Water Project, which moves water throughout California, if we knew that a listed species had now entered the area, that is truly would be a game changer for us. So it's so exciting to hear about the technological advances. I appreciate it. Thank you. Okay, and next up, Ryan, I'm going to ask you a question. But before doing so, just want to mention to the participants that we're going to turn to the Zoom question and answer function or Q&A function for questions. And so after Ryan answers his question, and so please feel free to ask any questions in that Q&A button. It should be at the bottom for any of our panelists. Okay, so Ryan, for decision makers interested in using eDNA and specifically the data produced from eDNA monitoring as a basis for regulatory or management decisions, but are new to this technology, what confidence can you offer in the reliability and robustness of eDNA data, and what potential concerns would you flag?
Sure. Thanks, Linda. I think it's such an interesting question because it bumps up against this sort of very human idea that we distrust information when it didn't come from our eyeballs. Like, if I didn't see it, how do I know it was real? And particularly in the regulatory context, you better be, if you get a piece of information in the world and you're going to make a regulatory decision about that or on the basis of that, you better be damn sure that your observation reflects reality. And with eDNA, because it's invisible, it's like, well, wait, like, where did that come from? Did you just make it up? Or, if I come to Assemblymember Papan and say, hey, I found an invasive species in your district, I'm guessing should say, well, are you real sure about that, right? Like, before we do anything. And so I've thought a lot about this, that like, what is it about invisible information that feels less trustworthy? Because like, if you think about it, we spend lots of time making decisions and interacting with the world in ways where we don't literally see stuff with our eyes, right? Like, if I see a plane, or rather, if I hear a plane fly overhead, my bedroom is in an attic in the SeaTac like flyway, right? So like, I hear planes all the time. I don't have to see them with my eyes to know that it's a plane that's there, right? Or and think about, like, if you're on a train track and you hear a train coming around the bend, you do not hang out waiting for that train to come around the bend just to verify with your eyes that it's about to hit you. You've got other ways of deciding that this is trustworthy information and using your other senses. And eDNA is another sense for the environment. And the cool thing about it is we get DNA sequences, so they can be really, really specific. We can, from a really short stretch of DNA, we can be really sure that it came from a particular species, which is really remarkable. In a lot of cases, we can be more sure from the DNA than we can with our eyeballs. So in a lot of ways, it's more sensitive and more specific than traditional means. And so then the second part of your question was, well, how do we develop trust in that? We're like, how do we develop reliability and understand robustness here? And I think people bring up a couple of questions where you can poke at it and say, well, are we sure about this? And one of those is false positives. And I saw John Darling here in the audience wrote this great article, like, what do you mean by false positives? And that's a cool example, because when we're looking at eDNA, it's an indirect way of seeing the world, right? So if we maybe don't have the animal in our hands, And so it may be that the DNA is somewhere nearby in space or time, rather than like directly on top of the thing that we detected. And so is that a false positive in the sense that it's like, is the animal wasn't there? And so that's kind of a question, but in general, like no, like we found the molecule. That's a real detection. The molecule was really there. And so then the question is, why was the molecule really there, or how far did it travel? And as Zach pointed out, not real far. So you find a molecule somewhere, it pretty much was there, and you got to grapple with that a little bit. The second thing that people have concerns about, where you could sort of worry that you got it wrong, is in putting a name, species name on the sequence. And so it is true, right, that sometimes, for example, dolphins are all very, very similar at, short stretches of DNA. And so it's true that sometimes you get one name attached to your DNA sequence, and maybe it's not quite the right name, right? Maybe it's a different species that's closely related. And so fortunately, we have ways of figuring that out, right? You guys can zoom in and say, well, how sure are we about this species name? So for things like invasive species or endangered species, that's what you'd want to do, is go double check and make sure you're happy with that name and it's not easily confused with something else. But again, I think this is just, to the point that Zach made, if this is just another net and this is a new net and we're learning to use it, this is like, how do we build trust in a tool? Well, we build trust in a tool when we know it works and when it delivers reasonable results, and when we use it in a way that is fit for the purpose, for which it's being used. And I think it's really exciting to have a chance to see this start to go into management and policy and get used in that way.
Thanks, Ryan. And I think that I really relate to those because for me, I have to work in a forum that is an interagency monitoring forum. And so it's a lot of different agencies that have a lot of different trawls going out almost every day of the year. And oftentimes we'll get asked the question about eDNA. Well, didn't pick up the exact same individuals that were picked up with the trawl. So do we really trust it? And in my mind, and I'd love to hear your thoughts on it. My mind, there is no tool that is 100% going to pick up every single thing that is present. There is bias to every tool that we use, and that's why they're great complements for each other. And that's why I like being able to use trawls, and I like being able to use eDNA and then be able to integrate those methods together.
I couldn't tell if that was a prompt. You actually,
Yeah, I would love to hear your thoughts. Yeah.
Yeah, and I mean, all of us on this panel, we've wrestled with this exact question a lot, and for a long time, it felt like, oh well, if the trawl gave me one answer and the DNA gave me a different answer, well, what's wrong with my tool? Like, why isn't it working? But it turns out that, like you said, right, every single method of seeing the world is biased in some way, and they're all biased in ways that are they're often independent of one another. So in fact, you would not expect those answers to match up. That both tools are a really imperfect view of the world, and where we get a better view of the world is by joining them together and getting a sense of, okay, these biases cancel each other out in a lot of ways, and then it's like, hey, the sum of them is going to give us the best view, and that's really what we're after.
Thanks so much. Zach?
Yeah, just piggybacking on Brian, I think one kind of fun recent California example that really talks to that we ran into is with our new sunflower star assay. So for folks that are not aware, Pictopodia helianthoides, sunflower stars, with the Pacific marine heat wave that hit 2014 to 2016, sort of went functionally extinct in the state of California. In fact, only recently, as soon as 2025, have we started seeing them in the wild, which has been really exciting. But we were doing some side-by-side scuba surveys versus intertidal sampling with eDNA and scuba sampling with eDNA. And one of the things we found was that intertidally, we kept detecting sunflower stars, but nobody saw one in the tide pools. And the answer was they were sitting, at 30 feet of depth, 50 feet from shore. And so if you were, trying to fit a curve of the biomass, against these things, you're like, well, it's the horrible curve because we keep detecting them eDNA. We're not seeing them. But it's really the scale of observations. If you take a step back and you say, well, what does the abundance start looking like within a region or within a bay or even within a cove, on the scale of a couple miles, things started looking a lot better when we started, comparing that. And so really the other thing about comparing these, observations is What is the scale that we're looking at? And, obviously with your eyes on a survey, you're often looking at, 2 meters by 2 meters, or, across 2 meters by 100 meters, for a lot of marine surveys is what you're looking at. eDNA, is looking at something probably closer to the size of a football field while we're sampling, and it's more integrated over space and time. And so we just have to be really thoughtful when doing those like side-by-side comparisons. They're actually doing a fair apples to apples comparison. And, at the end of the day, what really mattered, right, is where are these sunflower stars in the state, right? And now we have a tool that we can start answering them. And you start, so yeah, sure, on the scale of a meter, they're not very useful comparison, but on the scale of, where along 840 miles of the state of California's coastline can we find some of the last remnant populations? Like, we have a tool that's quite useful for me to answer that question.
Great, thank you so much. Okay, we have a question. It's actually a two-parter. And so I think for the first part, Ryan, if you could answer. So I'm going to start off by asking the question. eDNA has been praised as a breakthrough in biomonitoring, but it seems like there is still a lag in its integration into management. What do you think is the biggest roadblock?
I think it's twofold. I mean, in general, we talk about managers. I mean, I think administrative agencies, whether the state or federal level, have a lot of autonomy in terms of what tools they want to pick to answer the questions there, to deal with the non-discretionary duties that they have. So it's not a question of like, can you use eDNA legally, right? That's like, yeah, well, yeah. And in terms of standing up in court, it's, each time that's been challenged, it's been very few, but I found 3 in the literature. 3 times showed up in court and been challenged, and all 3 times sort of stood up to judicial scrutiny. And so, like in terms of is it legally acceptable? Yes. Do they have the legal authority to do it? Yes, no problem. And so therefore, I think the roadblocks really are twofold. Like, first, sort of cultural, I mean, that kind of I talked about before, like, is this a real thing? And that's starting to be, that's now starting to be kind of taken care of through for like this, right? And then there's this technological hurdle. I think it's like a know-how hurdle. Like, okay, fine, yeah, I'm in. I want to use eDNA sampling to do whatever job I have to do at the agency. How do I do that? And is that a service provider, or do we do it in-house? Like, Melinda, in your agency, you do it in-house, right? But not everybody is lucky enough to have that kind of lab facility. And so, and then once you get the data back, how do you interpret it if you're not a molecular biologist, you're not a statistician? And so I think those are it's kind of the last mile there of like, how do you get where you're going? And I think we're certainly getting there.
Excellent. And perfect segue into the next question, which I think I'm going to ask for Susie to answer as the lead for the molecular methods work group. eDNA data can be complex to interpret. What do you think the role of scientists is when it comes to translation of information?
Yeah, awesome. And I think this dovetails nicely with what Ryan was just talking about. So if I think back, a couple of years ago when we first started the California Molecular Methods Work Group, one of the big motivations was because we felt like eDNA tools were getting spun up in a number of different research groups or different agencies, but there wasn't this kind of coordinated effort to talk about like, how are we using these tools? What sort of methods do we like? What sort of, quality control measures do we like implementing to feel confident and really build that trust in the resulting data? And so then that same idea was echoed in 2024 when the White House Office of Science Technology Policy released the eDNA National Strategy. And then we also saw California Department of Water Resources has their own eDNA strategy. And the thread throughout all of those is like greater collaboration among eDNA researchers and practitioners, and then also better communication about what eDNA is and what it can do. Because we've really found that that's key to beginning to develop some of that capacity with the agencies that are interested in adopting these tools for long-term routine use. So I think scientists are key, but it's really about how they're collaborating with the eventual end users and helping build that technology transfer. Thanks.
Sorry, I keep muting myself and then I can't find my mouse to unmute. Okay, let me see. Don't see any other open questions. And so I'm going to ask a couple questions from the registration. Let's see, I'm going to Kevin, wondering if you could answer, how can eDNA be used to monitor impacts from offshore wind?
That's a great question, Melinda. And I think a bunch of us sitting here were recently at a offshore winds workshop in Monterey recently. And I think DNA offers a powerful sort of low impact way to assess biodiversity in and around offshore wind farms, like detecting broader range of species, including rare and cryptic protected taxa, than traditional trawl methods that might not be applicable in those spaces. Or other visual methods. So I think it enables us to sample in these sort of hard to sample places of offshore wind farms, but it also provides us a scalable way to do monitoring within the wind farms across the whole life cycle of offshore wind from, before wind farms are put in. So establishing those baselines, monitoring during when those offshore wind farms are in operations, and then the decommissioning of those systems. So it provides us this long-term ability to collect a lot of samples on the biodiversity around wind farms through the whole life cycle of these projects. And then from an instrument perspective, I think the only way to do that on the right scales is to use autonomous instrumentation, where we're not putting people in harm's way of sampling in bad weather or in perilous times or at night or those types of things where obstructions within the offshore wind farms can sort of impact the safety of folks trying to collect samples. Yeah, so I think I answered your question.
Yeah, you did. Thank you so much. All right, Zach, I was wondering, thinking about on a national scale for fisheries management in your role at NOAA, how do you think eDNA has been used in national scale fisheries management? And that's from both a federal perspective as well as California specific. And what's needed to further integrate quantitative eDNA techniques into fisheries management?
Yeah, happy to answer that one. So, most recently and notably, the Pacific hake fishery off the coast of both California, but it's a federally managed fishery, and also joint managed with Canada, has officially adopted eDNA as part of the science and data that's being incorporated in the stock assessments that are being used to actually manage and set catch limits for the fishery. And so this is the first time that this has happened. From NIMPS. I'm in NOAA research, so I'm just commenting on their the research and the work that they were doing to make this happen. But this really came out of work that Ryan is deeply involved in. Northwest Fisheries Science Center was really the lead on in being able to develop quantitative PCR assays for hake, and demonstrating that side by side the traditional surveys that were done by both trawls and acoustics, eDNA plays a complementary way to really reduce the variance, and it made the stock assessments more accurate. And so going through that really incredible research effort, a handful of peer-reviewed scientific papers, as well as the formal stock assessment regulatory mechanism. And so I think there's a lot of excitement, sort of within the agency. It aligns with a lot of executive orders that have come out, in previous, as well as the current administration, for how do we modernize and do, fisheries assessments, better, cheaper, faster. And so, I think there's a lot of excitement there, and it'll be really interesting to see going forward, how do we roll this out for every fishery, that is, in many cases, they're independently managed. And so it'll really be up to the fisheries management councils to make, those decisions. But where, we've seen a lot of value is, being able to increase replication, really get the variance down. So it gives us better answers. There's also a lot of, for example, untrawlable habitat. Like, we can't take surveys. It is not safe to take samples. So we actually don't have a good idea of, how many fish are out there. But we can take a water sample basically anywhere. And so, that provides us a lot of information over increased space, scale, depth. That lets us know, when and where, species are. And I'll say, a sister project, we do a lot of work, not in California, this is in the Bering Sea, but we deal with a lot of sea ice, and we can't get eyes on the water for 6 months of the year, in the Bering and Chukchi seas. And, those are some of our most productive fisheries. And, being able to know where the fish are, in January and spring are really important for making a lot of those decisions. And we don't really have another tool, out there than eDNA to take those kinds of samples. And so I think there's a lot of power in these tools to be able to do our stock assessments in the future. I don't know of any in state fisheries that have adopted or used eDNA. Would be really interested to know. But I think the Hake assessment really is the first one that has been done on the national scale, and I think really sets a scale of, hey, this is possible. It's giving us better data. And I think it really hopefully will make the next series of fishery stock assessments sort of easier to go through that process, because it was a long, speaking to the previous question, it was a long way coming, almost 6 years to incorporate that data into the official regulatory sort of mechanism.
Thank you, Zach. And I will say, from in California, one thing that we have done is use eDNA for Chinook salmon monitoring in, for instance, tidal regions, like looking for, are they using restoration areas, or how long are they staying in a particular system before they outmigrate, things like that. But when you were talking about the Hake example, it made me think about, oh, Chinook would be a great tool. The eDNA would be a great tool for looking at the Chinook fisheries management in the ocean as well. They're frequently at this point, they close the seasons in California because there aren't enough. And so in the last couple of years, the season's been closed. And so eDNA would be a tremendous resource to better understand where are they and when are they there. So yeah, I appreciate your insight. Ryan.
I just want to add quickly on that. The Hake example is such a great, large scale example. One of the things that I think has become really clear recently is like, it's really about efficiency. That, like, what I did not know that you had for many like traditional commercial fisheries stock assessments, you actually have to have a different kind of gear to count each kind of fish. Depending on what you're looking for, right? So that's like a crazy number of sea days and ships that are out there. But with eDNA, you actually it's just one. You could look at any species you want, and all of them, they all have the same DNA in the jar. So once you've collected the water, you don't need different gear types to look at 50 different fish, and you're going to do those stock assessments. You literally have that in hand, the sample in hand already, and you can query it and requery it, as has been mentioned a couple of times. And then to sort of link that to John Darling's question here in the Q&A, if I do think that fisheries is a good example of the kind of path to regulatory acceptance, that it's that you just need a few of these examples, right? Where now NOAA has said, okay, cool, it's useful for stock assessments. And we know that courts in a couple of cases have said, okay, it's useful for Endangered Species Act questions about saying what's occupied habitat and what's not occupied habitat. And I think you need a few of those before it's a little bit like the dam breaks. It's like, and in fact, all of us were at a workshop recently in Maryland where there were a couple of the big consulting firms, and I forget which one. I think it was Fugro maybe who said this. It was like, if it's good enough for NOAA, it's good enough for us. And so I think you get 2 or 3 of these decisions that feel like they're maybe piecemeal, and then all of a sudden, basically, that is oh yeah, this is just how we do business now.
All right, great, thank you. Want to get to at least one more question. Let's see. There are so many gaps in biodiversity monitoring in state waters. How can eDNA sampling and monitoring help fill those spatial gaps and gaps at depth that aren't monitored regularly? Let's see. Kevin, do you think you're the best person to take this, or does somebody else want to take a stab at this?
I mean, I can throw it in from mine. Susie might have some opinion on this as well. I feel like if we know where those gaps exist, we can use autonomous instrumentation to start surveying those gaps, and understand where those or fill in the gaps basically. If we don't have ship time or we don't have the spatial or temporal resolution that exists, then one way to do that is to sort of establish this backbone network of autonomous sampling systems to collect those samples on the spatial and temporal scale that we need. We just need to understand where those gaps exist in the data. That would be my perspective, but Susie might have something else to say about it.
No, Kev, I think you're exactly right. I think that's the vision. eDNA is a critical component of being able to establish those biodiversity baselines that we need to better understand how species are distributed across the state. And I think it's a cliched saying, but you can't manage what you can't measure is so true. And I think eDNA is core to us being able to actually measure the biodiversity up and down the state so that we can then see, where are some species potentially at risk? Where are management actions effective or not, and what are some additional areas that we may need to protect in the future. The one thing that I'm really proud of California for is we've really recognized that establishing this baseline is critical. And so the state of California has indeed funded this really ambitious project, the California Intertidal Biodiversity DNA Barcode Library, which maybe folks have heard about. It's an ambitious project that's kicking off. We have a kickoff meeting in January, but the goal will be to map biodiversity throughout California's intertidal region so that we really can establish that baseline so that we can equip managers with the understanding of who we're finding across the state and so that they can better evaluate where there are maybe biodiversity hotspots and when their management actions are being as effective as hoped. So anyway, that's another place where I think California has really been a leader in recognizing the importance of doing these sorts of things like filling in these gaps. And I think eDNA is really the key to getting it comprehensively done across California.
Yeah, and I'll finish this part just by saying one thing, which is that another thing that California is doing is they put the use of eDNA into our climate adaptation strategy. And so this is a document put out by the California Natural Resources Agency, and it is specifically mentions the use of eDNA for better understanding climate impacts to California species. So that's also although it's not all used ubiquitously yet, it is beginning to make way into very high level documents, which is tremendous to see. Zach.
Yeah, I want that's really cool. And one thing I want to link this to as well, this is something Assemblymember Papan had said, to kick us off here was, oftentimes we're very reactionary. And we start a monitoring program. For example, I'll pick on Cal Coffee after the sardine fishery collapsed, and we don't have any data from, when we actually had lots of sardine in the state of California. And so one thing that eDNA does, because we have this ability, like we talked about, to go back in time, but also, to revisit these samples that we've archived. And also it is quite cost effective. It actually gives us opportunity to answer the question that Mark had of how we do baseline environmental surveys. Like we can start taking samples now. And, instead of waiting for that next oil spill or, the next offshore wind project or the next invasive species to come, we can have those baselines. And even if it just means we're putting filters in a freezer, we can go back and we can say, oh, hey, here's the baseline, from this ecosystem, from this habitat, from before, insert whatever change we're trying to track. And so I think, that to me is a really interesting thing that eDNA opens the opportunity. It really changes how we, oftentimes, especially under our national laws, it's like, oh, we do the environmental impact assessment right before we're about to change something. But, if we're going to make a big change, to our, environment, we often need, especially ecosystem science, to have that really critical baseline data. And so, I think this really gives us an opportunity to start doing those, collection. And just as it's kind of a tangent, but just an example of, like we keep talking about, it's cost effective. What does this look like? We were talking about, it's about $100 ballpark. People give you different quotes and it gets cheaper all the time, a sample, right? And so if you wanted, for example, every single public school in the state of California, there's about 10,000 of them, to go out and pick, 3 habitats or something and take 10 samples across them, we're talking about a $10 million program to survey 100,000 sites in the state of California. That's unimaginable with any other method, to survey, 100, to get 100,000 sort of survey observations. Certainly not doing that, with person hours. And so I think that's what we're talking about in the scale of, this is a tool that you could monitor every mile of the 840 miles of coastline within a year if we wanted to. Like this is, within the feasibility of something that's possible, that, there was never enough scuba divers ever, to be able to do that many surveys in the state. And so I think that's really what we're talking about to answer this question of, how do we fill these spatial gaps and places that are monitored regularly, and it answers one of the questions that Tony had brought up in the chat about, community science. We can train people to take these samples. Obviously Kevin's on the one end of the scale making these unbelievable sensors, that are autonomous and can take samples, at the bottom of the ocean. But it's also pretty low tech. I mean, you can use a syringe and a filter, and we've done community science work with 7-year-olds in Malibu to take eDNA samples inside and outside of a marine protected area, and it's as easy as filling a bottle with water. And so it also opens the door to be able to do these kinds of surveys in a way that we historically haven't thought about. How do we collect data for state and federal agencies? And so I think there's some really creative ways in the future that we can use eDNA to really change sort of how we do business and sort of understand how biodiversity is changing over these incredible the size of something, the state of California, and the size of something, the entire West Coast.
Yeah, definitely. The scalability of the technology is unprecedented from what we've previously seen. So I want to move us into our final question. Would like to ask one final thought from each of you. If you could have decision makers take away one thing about environmental DNA, what would it be? And I'll get us started. I would like personally for decision makers to realize that we need to work across agencies within California to really integrate this into the way that we do business for agencies to answer our key management questions, and that will take trust by decision makers, a lot of interaction, but that I believe that we can start getting there. And so that's my hope for the future. Let's see, Susie.
I love that, Melinda. Okay, I would say my one take-home message would be that eDNA science is not new, and we now have decades of research that supports its strengths and limitations and how it can best be used to help inform decision making. And so my real dream is that folks understand there are many ways to use eDNA. You can use it to trigger follow-up visual confirmation monitoring. You can use it to close beaches or shellfish beds. Or turn off water pumps. So I hope that we can find a way to help equip managers with the knowledge and the know-how so that they feel confident in making those decisions about when eDNA works for them and how to best incorporate it into their long-term monitoring. Thanks.
Thanks so much. Brian?
Well, Susie just covered most of the waterfront, so it's hard to know how to answer that question. But I think the take home is that as decision makers are always going to reach for the best tool for the job, or should, right? For any given job, what's the tool you're going to reach for? It's the thing that you trust, and that's going to do the thing, how it works, and it's going to do it at a reasonable cost, right? It's sort of efficient and does what it needs. And so now eDNA is a very good tool for many cases, right? And not every case, right? And so I think that's my take home, is use the best tool for the job, but a lot of the times eDNA is a really good tool for whatever job you're looking at.
Great, thank you. Kevin?
Thanks, Melinda. Yeah, so one takeaway message for me for decision makers to remember, I think I'm sort of going to flip my hat towards all the instrument developers that are out there from a commercial instrument developer perspective, and say that like environmental DNA, for it to become truly impactful when we scale it, I think there's an investment that needs in that growth that supports people just as much as it supports the science. So supporting people as much as science, that scaling the eDNA. And I think John Darling is sort of getting at this with his question in the chat right now that I think Ryan is sort of typing an answer to. But that scaling eDNA creates more consistent, accessible, and timely biodiversity information while reducing the reliance on all these operational costs for traditional surveys, ultimately making ocean monitoring safer and more efficient. But just more importantly is that every investment in eDNA methods, tools, and workflows fuels a bigger growing community of like small businesses that are developing these tools and instrumentation. So small businesses, engineers, and innovators who are sort of building these next generation ocean technologies. So we're not only protecting our ecosystems and the science behind that, but we're like strengthening the people and the companies who are making this progress possible.
Well said, thank you. And then Zach, you get the last word.
All right. Well, the one thing that I just wanted to add, building off of Kevin's point, I think investing in the people and investing in the resources is really critical. And I think, the state of California is already doing that. And sort of one shout out example is with the California Intertitle barcoding initiative. And where I say that, having these reference barcodes makes it so easy to actually go out and makes more trustworthy data from your DNA, but it makes it easy to design new assays. And so, with the Sunflower Star one, it took us a year to get the reference sequences for all the sea stars from all the museums and putting out emails to collections and things. And it took us 4 hours to then design one once we had all the reference mitogenomes and order them and then get it working in the field, which is crazy. But that's the kind of thing that's possible when, we do these investments, sort of upfront. And so I think, like any new technology that we, adopt, we have to build something that's fit for purpose, and doing that kind of investment up front, whether that's on building reference barcodes, whether that's training people or building these systems, or investing in the new sampler that is going to be able to provide the information, in the same way that we've invested a lot in our weather satellites to be able to provide us this information on weather. One satellite costs $1 billion, but it's there for 10 years, and it's providing us information every second all the way around the globe. And so those kinds of scale of investment, I think, in technology and in people and in the sort of resources can make this go a long way. And so sort of leave the academic realm into sort of the operational realm. I think answering some of the questions in the chat, and what Kevin's saying is thinking about how do we actually take this to the next level, outside of one lab, and start putting into practice, for all the different amazing use applications we've talked about here today.
Thanks so much. And now I'm going to pull it back to Tanner. Take us away.
Yeah, thank you so much to you all for all your time and expertise on environmental DNA, on this really interesting chat about its use in ocean management and how we can make more and better decisions. Thank you to all those who watch, the registrants who ask questions, and those who ask questions in the Q&A function. I want to again offer our thanks to Assemblymember Papan for our sponsorship today and for joining us this afternoon. The recording will be posted on our website. We'll put the link in the chat. And please get in touch if you have any questions or follow-up. I will share our panelists' information on the screen. And just thank you everyone for joining.